last two classes we spoke about lipids and membranes. Now what we are going to consider today is how we have the transport from the inside of the cell to the outside or vice versa. So it's basically membrane transport that we are going to talk about today. Now if we look at the fluid mosaic model that we discussed in the last class, we know that this lipid bilayer that comprises the membrane is interspersed with proteins and channels that allow the transport of ions from one side to the other, whether it's from the inside or to the outside or from the outside to the inside of the cell. Now this is extremely important for cellular processes that are going on, and you also realize that all types of molecules will not be transported into the cell, not only because of their size but also because of their polarity, because the lipid bilayer has a polar head group on either ends, but it is interspersed by this hydrophobic tails of the lipids. Now because it is interspersed by the hydrophobic tails of the lipids, we have specific proteins that are going to allow the transfer of ions and molecules. But before we go into that, we will see the nature of molecules that are going to be transported. First of all, we consider those that are permeable to the membrane and those that are impermeable to the membrane. For example, if we look at small non-polar, hydrophobic, uncharged molecules, small polar molecules, gases, these would be permeable to the membrane because they would generally basically diffuse through the membrane. But if we look at other molecules, larger polar molecules, for example, ions or charged polar molecules like amino acids and nucleotides, for example, adenosine monophosphate, AMP, ADP or ATP. Now these would be impermeable to the membrane because, as I mentioned, that since we have this hydrophobic lipid layer, it is unlikely that it is going to allow a charged molecule to pass through. So you have to have some channel or some way that the molecule has to get through. So considering what could happen, let's see what the transport, some of these features are. First, the lipid bilayer is practically impermeable to water soluble, that is hydrophilic molecules. The reason again being because it's a lipid bilayer. It would not allow this transfer to be favorable. Second, what cells actually do is they import water soluble nutrients like sugars, amino acids, they eliminate waste products and control ion concentrations. Now this is extremely important in maintaining what is called a sodium potassium balance in our cells. So this is what the cells actually have to do, and to do this in the importing of the water soluble nutrients or the elimination of the waste products, that has to be done through these channels. So what you have to have is, we have, as we have studied, we have integral membrane proteins, we have peripheral membrane proteins and we have anchor proteins. So what happens is, there is a specific requirement for membrane transport proteins that have to span the membrane to facilitate exclusive entry or exit of specific molecules. Okay, for the specific transfer or for the specific action of the cells, there are these specific transport proteins, and because each of them have a specific molecule that they are going to bind and transport, each membrane transport activity is unique. Now each type of membrane therefore has a characteristic set of transport proteins which will determine what is going to enter the cell. It is like a security control. Each of these membrane proteins will exactly determine what type of ions or molecules are going to enter the cell and when they are going to enter the cell. All this is controlled by these transport proteins. Now the membrane transporters, that are these proteins, are actually of two general classes. The two general classes are carriers and channels. Okay. Now what carriers do is, actually in a carrier mediated solute transport, because of the lipid bilayer, it is unlikely that this specific solute will be allowed to go from one side of the membrane to the other solely because of the lipid hydrophobic tails that are present here. So, what happens is there is a specific carrier protein that cycles between conformations which allow the solute binding on one side and release on the other side. Okay, so what actually happens is there is a specific conformational change that occurs, bringing it into a fashion that will allow an opening on one end of the membrane only. So, it is not a channel. A channel is something that would be through, all through. So, this is what you would call a carrier protein. This therefore carries the molecule from one side to the other, and in doing this it has to have a conformational change that occurs only on the ligand binding. So, once the ligand is bound, what happens is it changes its conformation. Then it sort of a reaction is instigated where the molecule is then eliminated on the other side of the membrane. Then to come back to this, it probably takes in a waste molecule from the cell and then changes its conformation again, then goes back to where it started from, exactly like a normal enzymatic mechanism. You know that it goes through a certain mechanism where there is a certain change, and then to revert back it has to do just the opposite of what it had done. Okay. So, this is how a carrier protein would cycle between conformations in which the solute binding would be accessible on one side of the membrane or the other, and there is no open channel all the way through the membrane. Now, these can be of different types. They can be what is called uniport, symport or antiport. Okay. Now, what this means is you have, I can show you the diagram here, it will be easier. In a uniport transporter, you have facilitated diffusion that takes, so A is going, so it is going from one direction to the other. That is uniport, it has just one directional movement. Symport has bidirectional movement. Okay, so it is co-transport, it transports A and B together. So, it is a synchronous process that occurs when you have a symport. In antiport, you have an exchange diffusion. You have A going in one direction and B going in the opposite direction. Okay. So, the classes of carrier proteins, these transporters are either uniport, symport or antiport, and uniport means that you have just the transfer of one set. Symport means you have the simultaneous co-transport of A and B together. And antiport means you have exchange diffusion where A is going in one direction and B in the opposite direction. Now, we look at ion channels. Now, what ion channels are is we have basically openings, through openings, but these are also sometimes what are called gated ion channels. Gated means basically it has a gate. Okay. So, it opens and closes depending on concentration differences inside the cell and outside the cell. So, whether the cell needs potassium or sodium would depend upon whether this ion channel is going to open or close. Okay. So, the channels basically cycle between open and closed conformations, and when open you recognize it is not like a carrier protein, where a carrier protein does not allow anything else to get through. Once this channel is open, there is going to be a flux of many ions going through because you have an open channel now. Okay. So, this channel is actually very sensitive to the flux. So, what is going to happen as soon as there is too many going through, it is just like a security gate, as soon as it sees too many ions going through, it just shuts itself. Okay. This is unlike the carrier protein where you recognize that once the ligand is bound, then what happens is there is a conformational change, but nothing else can get through. But when we have this open channel, it forms a continuous pathway through the bilayer. So, it is open for everybody to get through. Okay. So, all the ions will just try and push themselves through, but the cell might not require those ions. So, then it will shut the gate so that it is not allowed through. It is a beautiful way that these membranes actually work. And gramicidin is an example of such an ion channel, the structure of which was solved quite a few years ago, which is a combination actually of D and L amino acids. This is one of the proteins that not only has L amino acid, but also has D amino acids. So, what we find is a conformational change in this case also, but the cycling is between an open and closed conformation. So, the differences between what you would have a carrier protein and an ion channel is quite clear here, right. Okay. Now, how does this transport take place? There can be diffusion, okay. There can be osmosis, there can be what is called facilitated diffusion, okay. Let's go one by one. When we consider diffusion, it's just like small molecules that pass through the cell membrane, okay, just generally pass through, because there's just enough interstitial space or interspersed space, but you see that none of these are charged in that sense, okay. So, they just pass through the cell membrane, and the membrane is basically permeable to these molecules. So, it's just an ordinary diffusion. Now, you recognize that this is essential so that you have the CO2 O2, because when you have oxygen bound to hemoglobin, right, or the CO2 bound that has to be released, it has to be a normal diffusion in and out of the cell. Osmosis will allow the movement of water flowing from a region of high water concentration to a region of low water concentration, and I am sure all of you know what osmosis is. Facilitated diffusion is something that carrier proteins do. What they do is they help the diffusion of these certain proteins or certain ions or glucose or amino acids or whatever has to go through the membrane. It helps. So, it is a diffusion, but it is helped or facilitated by the presence of the carrier proteins. Okay, so, we have diffusion, we have osmosis, we have facilitated diffusion. We also have two other kinds of transport that are called passive and active modes of transport. Okay. Now, when we have passive transport, it means that there is no extra energy required for the transport of the solute from one end to the other. Now, when we are speaking of energy, okay, we are speaking of the free energy. So, it is a spontaneous movement of the solute molecules from one side to the other, be it inside to outside or outside to inside. That is what is called passive transport. When you have active transport, it means you are going against a gradient. Okay? If you are going against a gradient, you have to push your way through, and in pushing your way through, you have to expend energy. Okay? That energy comes from ATP hydrolysis. It is coupled with, we will understand ATP hydrolysis and where this energy is all coming from when we do bioenergetics. But for now, we have to know that when we have this membrane transport, we have two kinds of transport, where we have passive transport that does not require any energy for the solute molecules to pass through, and active transport where the cell has to spend energy, and the place where it gets this energy from is from ATP hydrolysis. ATP hydrolysis, there is a high energy bond that breaks and energy is produced, and it is beautiful in the way that there are two reactions that are always coupled together, where one reaction needs the energy and ATP supplies that energy. It is not that ATP will just break up to provide the energy if it is not required. So, essentially for both cases, you understand that this delta mu, that is the free energy change, has to be less than 0, because it has to be a process where the spontaneous transport of the solute molecules are going to occur. Okay? In the case of passive transport, this extra energy is not required, so delta mu is less than 0. But for the active transport, where one of the reactions is going to have a delta G that is positive, the energy released from ATP is going to more than compensate for that delta G positive value and give you an overall delta G that is negative, and this will be coupled, therefore the two reactions will be coupled to give you an overall delta G that is negative, so that the transport can occur. Okay. Now, if we look at the thermodynamics of membrane transport, we are speaking about free energy and how this all can occur. Okay, so basically we are looking at two chambers here where we are transporting one solute, say, or one substance from one side to the other. Okay. Now, if we look at this generally, we have, say, just general membrane transport where we are looking at, say, a box here where we have a membrane and we are looking at a region, say, alpha going to beta. Alpha is a region of high concentration and beta is a region of low concentration of, say, a solute X. Okay. So, now when we write the chemical potential, we have mu x which is equal to mu 0 x plus RT ln x in general. Okay. Now, if we have this going from A to B, then we have to look at the change of mu x B, because that is our final state, minus mu x alpha, which is our initial state. That is going to give us our delta mu, say. Okay. So, if we write out this for beta and we write out this for alpha, the delta mu that is associated with this is going to be RT ln, because what happens is this cancels out because we are speaking about the same solute X. So, we have RT ln the concentration of X in the beta low concentration side and the concentration of X in the alpha side. Now, we know that the concentration of X at the beta side is less than the concentration of X on the alpha side. So, what can I say about delta mu? It is negative. Okay. So, what do I have here? I have a spontaneous process that is going to allow the transport of X from the high concentration side to the low concentration side. Okay. This is something you have done before, but it is just basic thermodynamics that all of you have studied. But what we are looking at is we are looking at a normal spontaneous reaction that is going to occur here, because delta mu is negative and we have the transport of X going from alpha to beta, where the concentration of X in alpha is at a high concentration and at a low concentration. So, this is similar to what we have here, where we are considering the concentration of the component C, and we have again the chemical potential calculations where we can then determine what the transfer delta mu is going to be from 1 to 2. So, it is going to be mu 2 minus mu 1. Again we have RT ln C2 by C1, and if you put in the correct value of R, you are going to get your delta G value in joules or kilojoules per mole. Now, if the concentration of C2 is higher, then this is going to be positive. If the concentration of C2 is higher than C1, then we have a positive value for ln, which makes delta mu positive. So, the transfer from 1 to 2 is going to be unfavorable, right. So, what do I have to do in that case? If I have to have the transfer occur in terms of a membrane transport, I have to couple it with ATP hydrolysis. ATP hydrolysis is going to give me a huge amount of energy that is going to compensate for the positive amount that I get here. Okay? We will see that very clearly when we do bioenergetics, but for now what we need to know is for the transfer, if I have this C2 greater than C1, then my delta mu is positive. So, the transfer is not going to be favorable. However, if C2 is less than C1, then my ln is negative and my delta mu is negative. So, the transfer of 2 to 1 would occur spontaneously because I have a negative. And then when do I have an equilibrium? When I have the same concentrations on both sides. but this does not happen in membrane transport. You do not have continuous diffusion till the levels of the concentration are the same on both sides. For example, we will be considering the sodium and potassium levels in the cell, which are, they never get to a constant level. If they do, you will be seriously in trouble with your health. Okay. So, let us consider what we have here. Now, when I speak about a sodium potassium channel or a sodium potassium transport, you understand that it is transporting ions now. Now, when I transport ions, I do not have a free energy only associated with the transport due to the chemical potential. Not only a concentration difference, I also have an electrochemical potential associated with it. Okay? So, what do I have? The free energy change is going to be associated, you know delta G, you know Nernst equation. Delta G has, with an electrochemical potential, you have it equal to minus NFE. So, not only do you have the potential due to the concentration difference, you also have a potential due to the electrochemical difference when you are transporting ions. Okay? So, when we have the movement of these charged species, it will give a rise to the potential difference across the membrane, right? Now, due to the potential difference rise, there is going to be an imbalance of ion concentrations across the membrane. Okay? So, we have to have an electric potential. Now, we know, now this is just this basic definitions which you all know. You have a Z. What is a Z? Z is the valency. You have Na, which is the Avogadro's number, and you have E, which is the electronic charge. Okay? And we have this F. What is F? F is Faraday's constant, that is Avogadro's number into the electronic charge. Okay? So, basically what we are saying now is, when I do not have just a concentration difference, but I also have the transport of a charged species, I have to consider an additional electrochemical potential. So, what do we do now? So, we have a membrane with a potential difference across it. Now, I not only have, if we look at this expression here, what does this expression have? It has an associated with it a concentration, because of the difference in the levels of concentration of sodium and sodium in side 1 and side 2. But since we have a charged species now, what do I have? In addition, I have an electrochemical potential, and this is also going to contribute to my delta mu. Okay? So, I have now a system where at Na1, say, this is the concentration of the sodium at side 1, it is 145 millimolar. The concentration at side 2 is 12 millimolar. Now, when I consider the delta mu, I also have to consider, so where am I? I am transferring from 1 to 2, right? So, let us go back to this just to make it clearer. So, I have now, let us say, I have my cell, and here I have chamber 1, here I have chamber 2. In chamber 1, I have the concentration at 145 millimolar for sodium and the potential V1. In chamber 2, I have 12 millimolar and V2, which is the potential. So, if I am to calculate the delta mu value, I have to consider mu 2 minus mu 1. So, the contribution is going to come from RT ln, what is it going to be? Na plus at 2, because that is our final state, Na plus 1. Also, I have Z F V2 minus V1, the delta V that I have. So, if I calculate this actually, I am going to have, what is this? It is 12, 145, both are in millimolar, and I have a Z and an F, and this V2 minus V1 is actually 70 millivolts, minus 70 millivolts. So, we have this. Okay. Now, if I calculate this, it works out to be minus 13,000 joules per mole. So, what is it saying? That the transport of sodium from this higher concentration side to the lower concentration side is a favorable process. And you have a process that is a concentration that you have to consider and the potential that you have to consider. So, these are the two considerations that you have to make when you are transporting a charged species through the membrane. So, what we have is we have a delta mu, that is mu2 minus mu1, RT ln Na2 by Na1, that is a concentration of the ions on the both sides, plus ZF V2 minus V1. When the system is at equilibrium, we can write this, which turns out that this V2 minus V1 is 67 millivolts. Now, this is very important. When we consider the ionic species in the cell, the concentration in the cell and the concentration in the tissue or in the blood, if you notice, the concentration of potassium in the cell is high and the concentration of sodium is low. In the blood, it is just the opposite. Now, this is an extremely delicate balance. You can have severe brain damage if this balance is even slightly distorted. Okay? If your sodium level in the blood goes, even if your say potassium level goes down to 3 millimolar and your sodium level goes down to 120 millimolar, you will have to be administered saline to get and be given potassium chloride to get your levels back up. And you cannot do that very drastically because your brain will be irreversibly damaged. It is extremely delicate. So, even this 4 millimolar cannot go down to 3 millimolar. This cannot even go down to 120 millimolar. Okay? It is that delicate balance, and this is extremely important because we have a specific carrier protein that can act on sodium and potassium. So, what it does is it brings the sodium out of the cell, and it is the same protein that will take the extracellular potassium into the cell. It is quite smartly, very, very smartly designed. So, if we consider these mammalian cells, they are selectively permeable and they open and close channels for particular molecules when they are required, as I mentioned before, because you cannot have too much sodium in the cell. You cannot have too much potassium also. Too much is also bad. Okay. Now, in a resting cell, most of the potassium channels are open. Why? Because it can have a large amount of potassium in the cell, not outside. So, what do we have to have here? We have to have an ion gated channel. Okay. The ion gated channel will, in this case, what happens for the resting cell, most of the potassium channels are open so that the potassium ions can come through, but only a few of the sodium channels are open because we do not want too much sodium in the cell. Now, if we look at some interesting features of the sodium and potassium ions, then what we have here is, if we look at the radius of the sodium and potassium ions, we have K plus ions that has a radius of 1.33 angstroms. We have Na plus ions that have a radius of 0.98 angstroms. Okay. Now, if we consider that the sodium ions are small, so it is likely that they are going to go through more easily, but that is not true. You know that when you have hydration, right, the hydration sphere is different for sodium and potassium, but when it enters the cells, it is stripped of this hydration sphere, because what you have to do is you now have to traverse this membrane. Now, there is a certain molecule that is very cleverly designed called valinomycin. It is valinomycin. This is kind of a circular molecule. It is made up of 6 amino acids. So, we have some amino acid chains that valinomycin means there are valine amino acids, valine side chains rather, that are outside here. So, what are the valine side chains? What is the valine side chain? It is CH, CH3, CH3. What type of a group is it? A hydrophobic group. So, here we have a hydrophobic shell. Now, this hydrophobic shell in here has 6 oxygen atoms. Okay? Now, these 6 oxygen atoms are spaced in such a way that this fits the potassium ion exactly. And because of this hydrophobic surface that you have, what happens? It can pass through the membrane very easily, right? So, the size is such that the interaction between the oxygen and the potassium charges, the ionic interaction is unique. Now, because sodium is too small, it does not sit here. It does not have the specific interaction that potassium has. So, it is not transferred as efficiently. Okay? Now, this is useful because the concentration of K plus in the cell has to be high. Okay? So, the fact that valinomycin actually binds K plus with a much higher affinity than sodium is useful for the transfer of the potassium ion to the cell. Okay? So, and the reason for having these specific amino acids, hydrophobic type amino acids on the surface, is so that it can just pass through the lipid bilayer. Okay? So, it can interact with these lipid hydrophobic tails and pass through. So, nature has designed everything. So, what we have here is we have in a resting cell the K plus channels open, where it is transported using this valinomycin also, but few of the sodium channels open. Now, the membrane potential, which I used in the expression that I used before for the sodium potassium, where we have minus 70 millivolt, which is why we calculated the delta mu. What was the delta mu we calculated to be? Minus 13,000 joules per mole. Okay. So, what was that due to? That was due to the sodium concentration and also the potential difference. What is this potential? It is a membrane potential we are talking about. Why do we have a membrane potential? Because we have different amounts of charged species on either side of the membrane. So, that is going to give rise to a membrane potential. It is also going to give rise to a concentration gradient, and in the calculation of the delta G, we have to utilize not only the concentration gradient, but also the membrane potential. And later on, when we study about ATP hydrolysis and the ATPase pump, we will have what is called a proton gradient. So, not only are we going to have a concentration change, we are going to have a concentration change, a membrane potential, and also a proton concentration gradient, which is going to give rise to a pH gradient. Okay? Now, that is also important. Okay? So, we are going to have then the delta G contributions will be from 3 levels: concentration, membrane potential, and pH. Okay? We will do that later when we consider ATP transfer. Okay. So, now this is what we have for the potassium. We worked it out for the sodium. This is what we have for the potassium. For the sodium, it came out to be 67 millivolts. Here it is minus 95 millivolts at 310 K, that is 37 degrees centigrade, which is normally where you would do all your biological calculations. The pH 7.4 and 37 degrees centigrade, which in some books and most of the books in where you will read biochemistry, the delta G0 has a prime to it. The delta G0 prime means that that is a biological standard. Okay? In the normal delta G0, what do you consider? 298 Kelvin. When it is the prime, it means it is 37 degrees centigrade, that is 310 Kelvin, and the pH is 7.4. Okay? That is what that prime means. Okay. So, what do we have? If we consider now our sodium gradient, what did we have? We had 145 millimolar. Where is this outside now? 145 millimolar. We know that the inside of the cell has high potassium and low sodium. So, the sodium gradient is such that you are going from the outside to the inside of the cell. So, the tissue fluid has 0 millivolts. The cytoplasm has minus 70. Now, where has this minus 70 come from? It has come because of the ion concentrations being different on either side of the membrane. So, the interior of the cell is negative and the sodium concentration is higher on the outside than the inside. The free energy is gained by opening the sodium channels and allowing the sodium ions to flow. Why? Because your concentration on the other side is low. So, if you are going from a high concentration to a low concentration, then that is favorable, right? We calculated that the delta mu is going to be negative for such a case, because the ln is going to be negative, right? So, if we gain free energy by opening the sodium channels and allowing the sodium ions to flow, but we know that the free energy comes from both the concentration gradient and the potential difference across the membrane. And that, as I mentioned, when we have an H plus case, we are going to have, in addition to this, we are going to have a pH gradient as well that has to be considered in our calculations for delta G. Now, what happens is, as I was mentioning, the sodium and the potassium sets are actually coupled together. So, what we have is, if this is our membrane, we have what is called sodium Na plus K plus. It acts so much like an enzyme that it's been given a name Na plus K plus ATPase. Okay? This ase suffix is usually used for an enzyme, but this sodium potassium pump acts so efficiently that this has been given this name. So, say this is occupying this region here. This is where our Na plus K plus ATPase is. Okay. What happens is, if this is the outside and this is the inside, what happens here, there is a certain gated channel here where we have and we have ATP hydrolysis, because this has to be driven. It is an ATPase pump. So, we have ADP. We will be studying all this when we do bioenergetics in more detail. Okay. Now, What has to go from the inside to the outside? Which ion has to go from the inside to the outside? What do I have higher inside? Sodium or potassium? Potassium is high inside and potassium is low outside, and the opposite for sodium. So, Na plus is low inside and Na plus is high outside. So, whatever is going out is Na plus from the inside to the outside, and what is coming in is K plus. Okay. Now, what happens is the carrier protein is such, or the way it works is such that it takes out 3 sodium ions and brings in 2 potassium ions for every cycle that it goes through, because it is built in such a manner. You understand that the K plus ions are larger in size. What is the size? The K plus is about 1.33 angstroms and the Na plus is 0.98 angstroms. Okay. So, 3 of these Na plus are brought from the inside to the outside and 2 of the K plus are taken from the outside to the inside, and then what happens is this is then ready to take the sodium outside again. Okay, because you have to get it back to where it started from, just like an enzymatic reaction. So, let us see how this works. Okay. Now, what happens is we have the extracellular side. What do we mean by the extracellular side? It is the outside. And we have the cytoplasm. What is the cytoplasm? It is the cell matrix inside the cell. Now, what is going to happen is, I don't know how this works this way. Okay, this is what that's the second picture. Anyway, so what we have here is the cytoplasm and the extracellular side. These are the 3 sodium ions. So, where is the sodium ion? Where are the sodium ions going? They are going from the cytoplasm. They have to be taken outside, because we do not need a large amount of sodium ions inside the cell. So, what is going to happen is these 3 sodium ions get embedded in this protein, okay, this carrier, and they fit in quite snugly at this point. So, the 3 sodium ions have come from where? They have come from the inside of the cell. They are bound here and they have to be pushed outside. Now, once the sodium ions bound, they sit in here, there is a conformational change that occurs, and we have ATP go to ADP, releasing a phosphate. So, what do we have? We have an energetic procedure that has to occur here, that is going to release the 3 sodium ions to the outside of the cell. Now, to have this protein back to where it is, it has to come back to this conformation. Okay, it is now an open conformation. It has released the sodium ions that were bound to it. So, in the next step, what happens is, from the extracellular side, it takes up 2 potassium. Okay. So, the 2 potassium then sit in this side in the protein and bring about such a conformational change that the potassium ions are released on the cytoplasmic side, okay, just like you would expect to happen, because you want this level, the K plus level, to be high inside and the K plus level to be low where? Outside. Okay. Now, this is ensured by, let us look at the previous one. What happens here is we have these 3 sodium ions. The 3 sodium ions came in from the cytoplasmic side and were released on the extracellular side, on the outside of the cell. But to get the conformational aspect of the protein back to where it started from, so it can accept another set of 3 sodium ions. What is going to happen? It has to get back to the same conformation, okay? Because what happens in these carrier proteins is you have a conformational change that occurs, and in this conformational change you have to get it back to its original conformation so that it can perform the procedure once more, because it has to keep on doing this, it has to constantly do this. So, we have the 3 sodium ions that go through and they are released on the extracellular side. Then you have the potassium ions that bind to the protein that are eliminated on the cytoplasmic side to maintain this. Now, this is extremely important, this Na plus K plus ATPase pump, as it is called, which is also known as a sodium potassium pump. Okay? It is also known as a sodium potassium pump. Now, what we have, this is extremely important in nerve cell signaling, which is why if you have an imbalance, your brain gets affected and you have what is called confusion. Okay? You do not remember things because of the imbalance of sodium potassium. You have you do not have proper cell signaling, which affects your brain. Okay? Then what you have in these cases is what are called voltage gated ion channels. Okay? So, in these voltage gated ion channels, what do we have? We have, when we have our lipid membrane, for example, in gramicidin, what happens? You have a, say, a single channel for gramicidin. Initially, it has part of the lipid membrane. Okay? Let us just put the gramicidin in a different color. Okay, and one of them is here. They have pores halfway through the membrane. Okay, something like that. Now, what happens is when it has to form an ion channel, what is the movement that can occur for these lipid membranes? You have lateral diffusion and you have flip-flop as well. Okay. Now, when lateral diffusion occurs, in this case, what happens is at one point there is a dimer formation. What happens is this channel, this is your gramicidin half and this is a gramicidin monomer. This is another monomer. Now, when there is lateral diffusion, at one point what happens is it forms the channel through and through. Okay? And then, because what is going to happen is, even though an ion may get in here, it will not get to the other side because the lipid hydrophobic tail is there. Okay? The same thing for this case. An ion can come from whichever way you are talking about, inside or outside, extracellular, intracellular. An ion can get through, but it will not come to the other side because there is no channel through it. But due to the lateral diffusion, at one point what is going to happen is you are going to get a channel. This is what you see when you form gramicidin. Okay, this is what is gramicidin, and gramicidin is unique in that it has, as I mentioned, D amino acids. In apart from D, it has, apart from L amino acids which are common, it has D amino acids as well. Okay. So, what happens in this case? You have the monomers that are present and it forms a dimer. What happens in this case then? You have an ion channel that will allow the flux of ions through and through. Okay. And why is this possible? It is possible because your whole membrane is fluid in nature. Okay. Because of the fluidity, because of the lateral movement, it is possible that you have these ion channels that allow the transfer of ions inside and outside. So, what we have studied today is membrane transport. Okay. Basically, what we understand is that for the membrane transport to occur, we have concentration gradient. Okay. We have specific types of transport, active transport that requires energy, passive transport that is just going to allow the permeability of the molecules, the solute molecules through and through, and we have specific molecules that have to have specific concentrations inside and outside the cell, as a result of which there is going to be a potential developed because of the charged species that have to be transferred, and we have the concentration gradient as well. And we went on to do some free energy thermodynamics, the thermodynamics of membrane transport, where we calculated what the free energy changes were. Okay? Thank you.